All right. Good morning, everybody. Today is Wednesday, April 11th, 2018, and this is Before the First Cup with Jules here on UCY.TV. Happy Wednesday, everyone. It is a chilly 36 degrees and cloudy here in upstate New York. So where do we leave off on Friday? We titled the show Brain Uploads, Connectome Preservation, Barcoding, and Consciousness. We had spent a lot of time looking, probably more time than I had planned, looking at the brain on Friday, and with the hopes of getting to designer proteins, which I hate to tell you, but it's going to be on Friday again. We're going to try to get to that. I came down with my friend's flu this week, so I'm not really feeling up to the task of doing a show today. But thank you, Camille. Camille sent me a video off of YouTube this morning titled Personalized Gene Therapy, and it is an interview with the CEO of a company named Ligandol that is utilizing protein-based nanoparticles to get CRISPR inside of humans, of course, to cure disease. It's a pretty interesting conversation. He even admits that it can be used for evil, but hey, don't worry about it. Let's give you some evil laughter and tell you that it won't be used for evil. So hopefully you will enjoy this, and it'll be a good setup for Friday's show. Everybody take care.
Welcome to Simulation. I'm your host, Alan Sakian. Now we're sitting down with Andre Watson. What's up?
Hey, Alan.
How's it going?
Thanks for hosting.
We're talking about Andre's company, Ligandol. We're talking about gene therapy. We're talking about personalized medicine. We're going to get into depth in all of that.
I was in Boston for like 6 years, from like the ages of 13 to 19, and for some time wanted to go into music school. Then I decided I really liked physics, so I went to RPI for physics for a year. Then I switched to biomedical engineering. Started looking at all the different landscapes of, what is biomedical engineering? Because really from just, how am I going to spend the next 20 years of my life sort of perspective, I wanted to be doing something more impactful than just, am I going to build an app for, food delivery or something? Not to say that's not great, but, like a lot of people are doing that, and we're still going to die, and that's kind of frustrating. So that's where I started looking at really the field of, biology as a medium for engineering, right?
Yeah.
And that's really what I like about bioengineering and biomedical engineering.
Yes.
A lot of it focused on, implants or bioinformatics. How do we deal with all the data from genomics or, MRIs, that sort of thing.
Yes.
But what really struck me is that amidst all of the data that we could possibly have around any number of things, having our genome sequenced, having blood tests all the time, we were still really bound by these limitations physically as biological creatures, in that we can't really do anything about that information for the most part. And so what struck me also is that we had all these tools, RNA therapeutics have been going on for 20 years, all sorts of different gene therapy modalities. Now we have CRISPR, but it still has the same problems in that you can have the best tools for reprogramming a cell, but you need to actually be able to get those tools to the right organs, the right tissues, and the right cells, and do so in a way that actually reprograms, an adult human, so you're not having to rip the person open every time you want to do something.
Yeah.
So that's really what motivated me to focus all of my attention on the field of drug and gene delivery. I took some really interesting courses on that when I was at RPI, and it did strike me that there pretty much were very, very few actual therapeutic implementations of that on the market, much less even in research labs, that were actually being translated into meaningful inventions. So that's kind of where Ligandle came from. I spent about 2 years at RPI as an undergrad just working on essentially delivering this precursor to CRISPR called the TALEN, another gene editing tool, that we had these really good talents for correcting the disease of these bone fragility mice, but we didn't have any way of actually delivering those tools into the mice. So what ended up actually working out quite well was these nanoparticles could be injected into the bone marrow of the adult mouse, and we deleted a gene with 50% efficiency across that whole bone, and we saw a thicker bone in the injected leg concomitant to that gene deletion. So that really sparked my interest in spinning out a company, because at the time we were seeing the beginnings of the CRISPR publications, 2012, 2013, but no companies that were actually solving how are we actually going to get CRISPR to specific cell types.
Yep.
So that's kind of the backstory.
You touched on so many important things there. Okay, so something that creeps to mind right away is you mentioned kind of what was happening in the industry, especially in terms of the amount of data that was. This is a big thing for data. We have so much data, especially on our body, but we don't know how to analyze it and make it useful to our lives yet, and we're trying to figure that out right now. And so you saw that in the making, and then you saw that you wanted to also figure out how to take in whatever the drug was, and rather than ripping up the human every single time, it's can we make it so that this drug can deliver the molecules to the specific cell or organ or tissue, and so that is Ligandle. Is what you're doing.
That is our mission. Our mission is to essentially know what the precise addresses are of the different cells, tissues, and organs, and actually build the delivery systems, the one-click shopping, send your packages to the right place.
How does one know how to get the molecule to the heart or to the tissue or to the cell?
A lot of that has to do with understanding the surface expression profile, the protein expression profile of a given cell. So one cell will have a different set of receptors and combinations of receptors on its surface versus another cell. Ligands.
So in each cell, proteins operate differently based on if that cell is a...
Tremendously differently, yeah.
Okay, because then the DNA is the same, but the genes express themselves differently.
Yes, precisely.
Okay.
I'll give you an example. Like if we're dealing with, a T cell, like a specific immune cell, T cells express a bunch of different markers that other cells don't express, right? So you're able to know that a T cell is different from this other cell.
So you can have that molecule go towards the cells that express a certain...
stickiness. Yeah, stickiness.
And then how does the molecule sniff out the stickiness?
So that's where, this is really what a ligand is.
Okay.
A ligand is a molecule that binds to another biomolecule or a molecule, right? So I'll give you an example that serotonin is a ligand for serotonin receptors. So you have, thousands of different receptors in your body, and different cells are presenting different combinations of them. What we do is we go through, millions of solution spaces of how do we make these ligands based on which proteins normally would interact with which proteins. And we dive in and we borrow that from biology. We borrow the native protein sequences. And then we latch those onto the genes and the gene therapy modalities. So you can couple the instruction to the address or the zip code, as opposed to hoping that both of those things will happen at once through like a small molecule when you take a drug, for example.
And the address and the you said the zip.
The address versus the instructions delivered to the address. Yeah.
The instructions and the address. What is each again in the case?
For us, the address is the cell surface proteome.
Which expresses a certain way.
Yes. The means for getting to that precise address is the polypeptide-based ligands, which are derived from natural proteins.
Okay.
And the instructions are the gene editing modality and or the gene expression modality. Okay. So CRISPR, TALEN, mRNA, plasmid DNA, siRNA, etc.
Okay. Got it.
And we essentially can do these massive combinatorial robotic screens where we just iterate through and find the most optimal formulation for sticking to a specific cell, coupled to whatever gene therapy modality we want, and then we just find what works best.
Okay, so then, you guys are on the side of the instructions. Is that right?
I would say instructions coupled to delivery. I believe that one without the other cannot efficiently.
But you guys didn't make the delivery methods.
We do. We develop all the delivery methods. Yeah.
As well as the instructions.
Yes.
But the delivery methods, you're not using the conventional CRISPR or.
CRISPR is the thing that will be inside the package. So you order your box from Amazon.
Yeah.
CRISPR is a thing inside the box that needs to be delivered to the right address and then carried to your front doorstep. And only then can CRISPR actually do something when it's in the nucleus of the cell that you want to target. Only then will it actually bind to the right sequence and cut it. So without that, CRISPR, just like any other form of gene therapy, if you inject it into the blood, it just gets destroyed and or doesn't enter any cells. So we developed the delivery systems. They're actually nanoparticles, but they're peptide-based nanoparticles.
Okay, the delivery system is a nanoparticle. Okay, so you're operating on the scale of like thousands or tens of thousands, or even nanos.
100 nanometers. Yeah.
How do you operate on that scale?
It's all very visual for me, kind of tumbly. You imagine basic interactions. There actually aren't that many interactions you can really exploit to form something at the nanoscale. You can couple things together covalently, forming permanent bonds between them. You can form kind of emulsions like water and oil having different phases and putting your drugs in like the oil phase, but then the water's on the outside. And then the third principle is using essentially charges to interact with each other. So we do a lot of charged-based stuff and figuring out how to get the sticky charged stuff to be on the inside of the particle and the ligands to be on the outside of the particle, and then coupling that to actually what happens when you get to the right cell. How do you get that cell to eat the particle? And how do you get in the multiple subcellular barriers, because there are many, how do you actually get the payload to the nucleus to perform the precise instructions that you want?
What is the answer to that?
Well, a lot of iteration, and we've developed this sort of multi-layered architecture that's almost like a multi-stage rocket, if you will. In that, if you're launching a rocket to space, you have multiple stages. Here, you have multiple stages in the sense that the first stage gets you to the cell. Then you enter the cell. The cell has something called endocytosis, where it basically will form this little vesicle around the receptor bound to whatever is on the surface, and it will literally pinch it off and pull it in. Kind of like a soap bubble forming from a larger soap bubble that's just able to close up again, right? A lot of very similar, actually, interactions to that.
I like the multi-stage rocket. That's good.
And then you shed a stage.
Yeah.
Next stage, it helps you inside the cell to target the nucleus. And then the final stage actually releases the contents in a nuclear-specific manner using enzymes that are specific to the nucleus. And we also try to program things like time-to-release characteristics and having control over that, where, do we want this payload to be released over a day or multiple weeks? And these are all the things we're trying to learn a lot about by these massively, high throughput, essentially iterative approaches to developing the most optimal formulation for a given purpose.
Okay, so then you guys write the instructions and make the delivery method.
Yes.
Okay, and then you've also analyzed the cells that you're trying to
Yes.
receive, that are trying to receive it, to
Yes.
optimize the approach of how to get there.
That's precisely right.
Okay, so now let's do an example of sickle cell anemia.
Yeah.
Okay, that seems to be like the one that is being
Yeah.
worked on, like the.
It's a great example.
Yeah, okay,
That's a great example. So right now there are a few labs that are.
And let me see if I'm right. Is it red blood cells have trouble carrying oxygen?
Yeah.
Yeah. Okay.
Yeah, great example. I'm really happy you brought that one up, actually. So a couple of
100,000
labs.
people?
100,000, yeah, in the U.S.
US, yeah.
Well, sickle cell is an interesting disease because you have it's a rare disease. You have 100,000 people in the U.S. who have it. As many as like 40% of people in North Africa and some Middle Eastern populations in sickle cell and the thalassemias, which are similar but not actually sickled, actually have that much. And in countries like in continents like Africa, you have this issue where essentially malaria. If you have sickle cell disease, you don't get malaria, or you don't die from malaria nearly as often.
If you have it,
Yeah,
you don't die.
it's protective. So, just as a kind of a bookmark,
Yeah.
we should be careful with that one.
Yeah, because then
Carrying
maybe you
Africa,
don't
yeah.
want to. Wow.
Yeah.
That's so
But
crazy.
here in the U.S., we can start piloting some stuff.
Okay.
A couple labs are making some really great headway. One of them is Matthew Porteus's lab out of Stanford. They're actually doing some of the first ex vivo, where essentially you have to take the hematopoietic stem cells that will eventually turn into red blood cells out of your body, and then genetically reprogram them to no longer have the sickle cell disease mutation, and then you reimplant those cells.
But then how do they tell the other cells to carry oxygen well again?
So the hematopoietic stem cells are normally these really rare cells. They persist with you for life. They can be anywhere between 1 in 40,000 of your cells, like in your blood and your bone marrow, all the way to maybe like 10% that will have some basic markers for being able to divide a lot. That they're basically stem cell subpopulations, right?
Oh, got it.
And so they can divide so many times. We're talking like thousands of times, like a lot, right? So you don't need that many. You just need, there's actually a protocol where you will condition the body to no longer carry its own stem cells after you take some of them out. You kill off some of the other ones so that the new ones can engraft and grow and proliferate. So what we're doing is a little bit different. We're not taking the cells out of the body. We're putting the nanoparticles into the body to target those stem cells and reprogram. So in the case of sickle cell disease, you only need to correct a single amino acid in that hemoglobin B gene. Other things are more complex, but actually they're about
One amino acid.
Yeah.
That's it.
Yeah, there's just one amino acid that's messed up in the whole protein.
There's a couple of these diseases that are just an amino acid.
About 6,000 of them, yeah.
Diseases that are just one amino acid?
Single genes, at least. They might
Single
be
genes.
a few amino acids, but they're single genes that could be corrected.
Whoa.
About 8% of the U.S. population has one or more genetic mutations, essentially, that correlate to a rare disease. And the majority of them don't have any therapies, actually. And then a lot of other stuff is more complex, right? So if we start talking about oncology and essentially, people getting cancer, that's less likely to be just a single gene that you were born with, and a lot of environmental factors and damage over time, and who knows what else, right? So that's another approach where actually immuno-oncology is really, really promising, and that's also something that we're focusing on a lot, which is essentially, can we reprogram immune cells to kill cancer cells based on genetically reprogramming them to have the correct instructions. And this is actually the first gene therapy that got approved last year in the U.S.
Wow.
Chimera by Novartis is it's an autologous T-cell transplantation where they currently will have to take these T-cells out of your body, and they basically use lentiviruses, which are like HIV that doesn't divide anymore and carries the gene insertion that you want, as opposed to inserting more HIV, right? So you insert these things called chimeric antigen receptors onto the T cells, which are basically like an antibody fragment on the outside of the T cell that will stick to a specific marker. So in the case of like B-cell leukemia, you can actually target the B-cell leukemia and kill it. Problem is that it will also kill your healthy B cells, but these are like terminal patients who don't really have much option as standard of care. Well, that's kind of the field, you know. You can do so much. If you can actually just deliver, you uncover essentially the full creative force of humanity for whatever we want to do.
Is this also able to assist with restoring homeostatic capacity, like youthful homeostatic capacity?
I really hope so. So that's something that I'm really interested in for a number of reasons, and I think that in showing that we can deliver on a cell, tissue, and organ specific level, these regenerative medicine approaches become extremely practical. Because take things like you had Aubrey on here, for example. If you want to target senescent cells and kill senescent cells off, you could start to do that. If you know that there's something unique about the surface profile of the senescent cell,
exactly,
you can deliver killing instructions.
And you can
similarly,
you're on the way to hopefully learn that.
Yeah,
yeah,
exactly. And we can use all the data that people generate around the world, and by focusing on, really the goal is, can we have a 2-day turnaround time for what genetic instructions do you want to deliver and where? At that point, it becomes, we're just playing with biology like we're hacking computers.
Yeah. I'm really excited for that. I know. The healthcare industry is so ready for this, and then there's all the complications around pharma and hospitals. There's all the complications around that as well that they're also behind on. And it's funny because AI and nanotech and all these methods of exponentially increasing technology are now influencing an industry that is kind of a little OG in some ways. And so now the healthcare industry is about to be torn into by these technologies, and it's going to expand. I wonder how it's going to work in a way that hopefully is able to impact more people. If there's so much money that's currently being concentrated in the orphan disease area of research, if we can figure out how to take that money and distribute better gene therapies across populations of people, there could be a significant savings in the amount of money that we spend on.
Big time. I think that's huge. I mean, look, I think the goal, if you just model this, if the cost of producing a gene therapy is essentially a trend that is just decreasing even somewhat exponentially, let's say. Which it is.
Yeah.
It's very, very inexpensive quickly. It's going to get really inexpensive quickly.
I love that. Yeah.
So what if it costs, let's say $1,000 to deliver any set of genetic instructions anywhere in your body, either temporarily or permanently?
Amazing.
$1,000.
Yeah. Amazing.
Your margins start changing a lot.
Yeah.
Because suddenly you can actually, you don't need to hike up the price of your drug to $500,000 or $1.5 million per patient because you have only a few thousand patients.
Yeah.
You can actually afford to really take on a super personalized medicine thesis, and people are actually getting access to this level of care. There's less reactive care like we currently have, where you spend weeks in a hospital bed, spending $2,000 a day on your hospital bed. Now it's you get an injection, you go home, and hopefully you're better. And I think there's a tremendous cost saving aspect to that. And oncology is a huge burden on also just society. I mean, solid tumors and hematological malignancies, also known as just liquid tumors, basically cancer as a whole, is projected to be, I believe, a $169 billion market by 2025 just in the U.S. So that's absurd.
So as we take all the money that we save from orphan diseases, and then if we can make it $1,000 to send any genetic therapy to any part of the body permanently or temporarily, that's going to usher in a new era of the way that we not only treat genetic disease, but also that we try and augment human experience.
Yeah.
Are you guys ready for that?
I think there's going to be a lot of stuff that needs to be figured out, and there are clear, ethical implications and a lot of things that need to be deeply thought about. And, we're trying to take it one step at a time in terms of defining, okay, these are the first sets of diseases that we're going to target. We're going to show that on a cell type basis, like if I can target a T cell or a hematopoietic stem cell, I can uncover all these therapeutic modalities. And from there, as the business grows, that's always something that I think it needs to be a continuous dialogue. I mean, as we're seeing with even just Facebook and this congressional testimony, you can't predict all these things. A lot of it, it happens live, and you need to just be ready to take it as it comes and adapt and do what ultimately is the best that you can.
Bioethics is very fascinating. I don't think we're ready for bioethics yet.
Yeah, humans are babies.
Humans are not ready for bioethics. What we are ready for is treating genetic disease. I think we are.
Start with that.
We can start with that. Okay, so we have these single gene mutations. That's an easier one to target.
Yeah.
Cancer cells can sometimes be maybe multi-gene mutation, but maybe even emit a certain characteristic that we can sniff out and go.
Very much so.
I really love that.
Nanotech's great for some cancers, not all. In some cases, not all, of solid tumors, you have something called leaky vasculature, where essentially the tumor grows fast enough that the blood vessels are a little bit leaky. So if you have nanoparticles that are nanoparticles, they actually will seep through and accumulate in the tumor. Yeah, and I think with these sort of approaches, if you're actually able to target different cells, you can start to play with multicellular, multi-organ system dynamics also, and because you're basically able to say, I can, delete or insert any trait that I want in any cell. So I think the more multimodal therapies become the most interesting. And I'll give you another example, even in like the neurodegenerative disease, Parkinson's disease, ALS, all these other diseases. The minority of cases are just a single gene, actually. So like Parkinson's is well known to have this one gene called LRRK2, and if it's mutated, you have a high probability of getting Parkinson's disease. So that's less than 10%. I think it's like 5 to 10% of people with Parkinson's disease. So then there are approaches that are quite promising for being able to address these multigenic implications of this. So, my good friend Katarina Vols is working on a company called Occam's Razor. And what they're doing is they're taking these massive data sets of Parkinson's disease from all these people and essentially figuring out, hey, like what is actually unique to this person that's driving their Parkinson's disease. And with gene therapy modalities that are non-viral and truly customizable, you can start to approach these diseases on a one-off basis and just say, hey, now these 7 genes are messed up, let's go in there and fix them. And no one's doing that right now.
What is the closest thing that people are doing to that?
I think the immunotherapy and the immunoengineering is the most complex set of genetic engineering circuits that I've seen so far. I think that stuff's really interesting because it factors in how do you actually program an immune cell to have affinity for a given marker, but also how do you program the signaling and the activation of that T cell so that it actually divides and activates and mounts a killing response. So people are programming the full circuitry.
Yeah. Oh, that stuff! I love the way you speak about it. It's just so enthusiastic, and it's also I think you're doing a good job of making it relatable. But it's really difficult stuff without any biological understanding. It's very difficult. With minimal biological understanding, I think you're tapping in a little bit. But then, of course, everyone that's watching that is totally biologically savvy. They're all over it. They know everything that you're talking about.
Should we give a primer on like the central dogma of molecular biology?
Yeah. Please.
So DNA turns to RNA, turns to protein. That's kind of the central dogma. It's the idea that you have this genetic code in your nucleus, and you have about 3 billion letters, and those letters get read out into RNA, which is the way that you amplify that signal because you have like one gene. You need to create a lot of copies of it, and then those mRNA strands they go through and essentially the endoplasmic reticulum, and they become proteins. Ribosomes and endoplasmic reticulum. The ribosomes actually synthesize
what does an RNA expressed from DNA, double helix, RNA, the chunk of letters, RNA? How does RNA even come from that chunk of letters, and then how does it get expressed into protein?
You have like polymerases that run along. You essentially have like your genome, and then it will be read out into RNA by RNA polymerase and a number of other. There are a lot of different like transcriptional enzymes. But basically, one of them is responsible for literally like spewing together the sequence that matches the DNA sequence.
Yeah.
And that will then be exported from the nucleus and then go through, and the ribosomes actually will have transfer RNA, where there's a single amino acid corresponding to 3 base pairs that will, as the mRNA is going through, they'll match up and they'll daisy chain together, and that's how you get a polypeptide that eventually becomes a protein. So that's the central dogma. That's like the classical biology, right?
DNA, RNA, protein.
Yeah. And nowadays, we're learning there are all these other, everything interacts with everything. So you have RNA interacting with RNA, you have proteins interacting with RNA, you have ribonuclear proteins, everything is everything. You have transcription factors, which are proteins that bind to specific DNA sequences and, cause genes to be read out more or less, and things like that. It's highly dynamic. But also, also in this beautiful way that I really love about biology, because it's all parallel, it's all happening at the same time, nonetheless it's coherent. Somehow we're conscious, and somehow also you can do very predictable things with biology. It has this stability and this coherence within it that survives amidst all that chaotic complexity.
Oh my God, the human machine is so beautiful and it's so complex, and then everything that you're describing about it is so many years of research by swaths of people that have really gotten deep into the human body and how it works. And okay, so let's see if I can go all the way back. All right. Ligandal. For ligandal, how is, let's see here, let's see if I can do this. We have the information, and we have it being sent, so the information is being sent to the address in the body. And the information is put in there through nanomedicine. So you're at this 100 nanometer scale of putting together the molecules that need to be put together in order to sniff out those specific cells. Okay, and then the deliverance method is injection or oral.
It won't be oral yet, but it will be eventually.
Injection. Yeah. So I get an injection of this nanomedicine, and then it enters in through my bloodstream, and then it sniffs out the cell that it needs to get to, and then there's the multi-stage rocket that gets it into the nucleus of this. Okay.
Yes.
And then that's where the DNA is, and that's where it's able to make the changes through either CRISPR or through the Cas9 protein or through the other ones that you listed.
Yes. Exactly. And so we describe ourselves as like payload agnostic, right? Because we are getting the package there. Payload agnostic.
Okay. I'm happy that I can at least have a 0.01% understanding of what's going on.
It's precisely correct.
I hope I can get better at this. As a science communicator, it can be really difficult. You're a very gentle and kind person, I'm telling you.
Thank you.
Because most people that, Aubrey de Grey does not give you time to be gentle or kind. He's like freaking running around being like, if you don't understand biology, I have no time. We need to go and get people out of pro-aging trance. And you're like, it's okay. Like, yeah, let's take our time. Let's get through this. And I think we got through at least enough to get that bare foundational level understanding. Okay. Let's go back to this information again. So the information that's going to the address, the information, besides being the right molecules that will sniff out the right cell, What else is being added to that as a nanomedicine and the multi-stages? You already said the multi-stages, but like, how do you know how to equip what is it, a hemoglobin stem cell?
Hematopoietic stem cell. You can call it an HSC if you want.
HSC, a hematopoietic stem cell. How do you program the nanomedicine to tell that hematopoietic stem cell to proliferate more and to take over or help with sickle cell anemia?
Well, that's a great question, and that's something that we're really pioneering also, because you touched on an interesting point, which is not just the reprogramming of the cell, but also the proliferation. So we are doing combination delivery also, and we were, I think, the first company to deliver like a gene editing, ribonucleic protein like CRISPR along with an mRNA, let's say. And we did these studies actually in monkey bone marrow, in cynomolgus monkey bone marrow, taking just the bone marrow and looking at the different subpopulations of like stem cells versus immune cells and so on that are eating up the CRISPR payloads. And, we're able to get it in. Like, we can get multiple payloads, multiple instructions in. So what does this mean from the perspective of how do you actually reprogram a hematopoietic stem cell to no longer have sickle cell disease? Well, there are two ways that you can do that. One is there's actually a gene that you can delete. It's kind of a hack. If you just cut in this one spot, it actually causes your cells to start producing fetal hemoglobin, which you don't produce after you're a fetus. But the fetal hemoglobin is able to rescue a lot of the oxygen carrying capacity. The slightly harder thing to do is to actually replace the gene of interest with the new sequence. It's easy to cut, it's harder to paste, and there's actually a delta and an error rate between those two processes. However, you can actually cut around the area of where that it's called an E7V mutation. It's the 7th amino acid, basically glutamic acid that is swapped for a valine, and you need to swap it back. So you can actually cut or perform some sort of obstruction of just the DNA, either a single-stranded break or a double-stranded break, and then you provide a template for repair. A couple of other approaches no longer require the cut and paste approach. So like David Liu's group at Harvard came up with a way to do essentially just replacing a single base. They call it base editing. And similarly, you would use actually like a modified Cas protein with the guide RNA that actually hones into the right section of the genome. And then you will actually metabolize one of the base pairs into a different base pair. So that's like more precise. That would be more precise when it actually works.
How many people right now are working on
11.
11 people, and it's mostly scientists.
Yeah.
Mostly in the field of nanomedicine.
Very multidisciplinary.
Oh, yeah, okay.
Super multidisciplinary. So like we have Xiao Lang. He was the first person to work with CRISPR. He was actually at MIT or the Broad Institute back in 2011, and he's like very good at genome engineering.
Awesome.
Then we have like Ryan. He's a peptide chemist. He was like at Lawrence Berkeley National Labs. He built us like a beautiful peptide robot that's like 60 times faster than anything money can buy, and then you just type in the amino acid sequence you want and it will spit it out. So you can see how, like those two things combined already kind of explains a lot of stuff. And we have people like who used to work at Genentech and stuff like that, working on nanoparticles and nanoformulations and chemical engineering, biophysics, biochemistry, material science and engineering, molecular biology, cell biology, genome engineering, peptide engineering, and also the AI, the robotics, the machine learning, the actual building of the processes for once you actually know the physical parameters of how to form nanomedicine, which is complex, but once you actually figure out how to make good particles, and you can start changing which segments encode for what, whether it be the gene or the protein that is actually targeting another protein, you can do a lot.
With automation as well.
Big time. It's all so automated. It's kind of the whole.
Yeah. That's so good. Okay, so you guys just did Series A, or how long ago did you do Series A?
Well, we raised a round last year, and we'll have an announcement a little bit later this year in June.
Okay.
It'll be some
Congrats.
big news. Thank you.
Good. Awesome. Who are some of the investors from the previous rounds?
Social Capital, Chris Larson from Ripple,
Awesome.
Esther Dyson,
great.
D.A. Wallach, co-founders of the Parker Institute of Cancer Immunotherapy.
Awesome.
We have probably 50 plus investors at this stage.
Nice.
Thank you.
Hell yeah.
Thank you guys.
Yeah. Yeah,
and super thanks to them. Would not be here.
And also super thanks to the vision and the execution and the whole team that builds it.
The team is incredible. Without a team, it used to be just 2-3 people, or Christian or CTO just literally being, let me get everything to work by myself, and I'm let's get us some money. It used to be the role. Now we can actually just work on all these pieces. It's really exciting.
How many clients are currently using the technology?
We're not on a client-based system at all. We're really developing the technology internally and doing all of everything up to the clinical scale and stage of manufacturing ourselves. So we're in the process of building out a facility that will actually allow us to do clinical grade and clinical scale manufacturing of all of this stuff. And it's going to have a robotic Ironman dome. I'll talk about that in 4 months. But yeah, it's pretty exciting.
So you're building out a whole facility. So you're doing the vertical. You're doing the vertical
and the horizontal
and the horizontal. But the interesting thing is then I will come into your clinic after you get FDA approval for this, and then I will leave your clinic having this $1,000.
And to set expectations, the $1,000 mark is when we're really personalized, and also we want to build clinics. That is the long-term goal.
You
for
do.
Sure. Long-term, I really want to see this where you can step into a clinic and get medicine right back, right? But in the interim, in this rare disease and immuno-oncology space, it's actually a very hot space for partnering and something that we're very interested in. Because also as a R&D focused organization, we don't want to take on all of the clinical development ourselves. We don't want to have to take on all the marketing and distribution and so on that's necessary to actually conduct a clinical trial and go to market. So we're focused on getting good data and then finding good partners to scale that.
Do that.
But because we have this foundational infrastructure and full supply chain and just knowledge of how to build the right technology for X application, that's where we can, we basically we enter the business of essentially venture creation and partnering and driving down costs and making things really widely available.
Yes.
And then once you're approaching that point where I can actually feasibly inject you with whatever instructions I want for $1,000, that's where that's where the whole model just becomes different, I think. And you might still actually lean. I think pharma will have a strong role. I think and I think pharma will continue having a strong role in just kind of this infrastructure, as it is evidenced by a lot of just the M&A activity in the gene therapy landscape across pharma, like Avexis, a gene therapy company that was just using viruses to reprogram spinal muscular atrophy in neonates or children, very, very young infants. They showed that they could correct this disease in 16 patients, and they just got bought for, I think, $8.7 billion in cash from Novartis. And that's phase 2 data, right? So I think...
Oh my gosh.
Yeah, and that's one application with promise. Maybe we can do some more things, right?
Yeah.
So what if you actually personalize it? What if it doesn't cost $150,000 per person? The first ones will. But,
correct. Yeah. Oof.
Yeah. Staging's everything. You have to time the market with the goal of doing social good and actually making things accessible.
Yep. Let's pull up your the email that you sent with the deck. Let me get connected to this, and then it's coming up. Because I think this deck looked...
It's a fun deck.
It's a good deck.
It's a good deck.
The scan converter's on, but I'm also a little slow on... Okay, there we go. Andre updated. Okay. Here we go.
Perfect.
All right. We'll go up until 4. Are you... you seeing it, Ron?
No.
Oh. All right, let me quit Scan Converter quick. All right, I just turned it back on. Yep. All right, so here's an investor brief for you. All right, so genetic engineering. You can have the fanciest ideas and molecules, but if you can't get them into the cell, they are of no use. Yep. And that's getting it from instructions to the address. I like that's so relatable. I really like that. Yeah. Growing understanding of genetics and immunotherapy, billions of dollars invested into CRISPR and other gene therapies, we lack the delivery mechanisms necessary to genetically reprogram diseases in ill humans. The biological equivalent of the last mile. Ligands. The gene therapy is exactly where they need to go. Biomolecules that stick to the other biomolecules, binding to precise receptor profiles of targeted cell populations. In vivo gene engineering, super personalized medicine, single IV injections, biggest development in medical history. Boom. I like it. I like it.
That's the dream.
I think we're 10 years away from this sort of an interface where you're just like, you're strapped in and I'm looking at your actual, visualized your actual DNA and I'm like, oh, you want this? You want that? Okay, great.
Well, you can already go to like simplygo.com and define a target genetic locus and get a CRISPR design that will target that sequence in 20 seconds. So we're already pretty close. We're doing pretty well.
Wait, what?
Yeah, it's like $50.
Whoa.
Yeah, it's a
So
thing.
you can get the information that you need to, but then the mechanism of delivery.
Yeah, you can get it actually delivered to you. Not delivered. You can get Synthigo will literally ship to you the guide RNA that targets the right genetic locus, right?
Whoa.
And you couple that to the Cas9 protein. Getting it into the specific cells of interest is where we kind of kick in. Yeah.
Yeah.
And those two are going to be, I think, very much like that.
Damn. Okay, what else shall we, about ligand, what else is really important to talk about before we get into our simulation question?
Oh, we're going to talk simulation.
Yeah,
I like that.
Yeah.
Ooh, I'm excited for simulation questions. I think we've covered a lot. I mean, I think this idea of creating super personalized, even multimodal therapies that can cover the whole gamut of health and human disease. It's really a matter of we need the zip codes, we need the addresses, we need the delivery mechanisms, and from there, all of the stuff that anyone is doing around the world, trying to, gene edit or, engineer a circuit of genetic activity or some sort of immunotherapy, like all of that just becomes kind of one. Just like we don't view, our computer keyboard as different from our monitor or from the computer itself. It's all, you can type. You can delete, you can insert. It's all seamless. It's not disjointed. But 40 years ago, it was. We didn't have a UX.
Mm-hmm.
So it's creating that and implementing it and making it so that it's not, a bunch of scientists spending 16 hours a day with pipettes, but rather people actually thinking and kind of pushing the direction of the evolution of the technology.
Mm-hmm. And then there's like that pipetting robot from Opentrons as well. I thought that was really interesting for the industry.
We have one.
You have one?
Yeah,
you have an Opentrons?
We have two, yeah.
You have two Opentrons in your office?
Two, yeah.
That's so funny.
Yeah.
Will, right?
Yeah.
Will, shout out. Andre.
Shout out to Will.
Yeah, okay, since this is our topic of the day.
I'm excited.
Let's talk about this then. How will we work together across the world, as for example China's AI budget is now the largest in the world. The genetic engineering we're hesitant, but they're running with it. They're trying it out. They want to play it. What do you think about that? How do we work together as the world to hopefully not create biowarfare or any other issues that could arise from you injecting somebody that just goes and kills their heart or whatever? That already exists, but
well, the pessimistic optimist in me, I'm not going to say I'm not going to say any bioterrorism ideas today. Kind of avoid that. But, I would say, if you really want to kill someone, polio or smallpox works, right? Like we don't need. I think there are all sorts of crazy ways that you could use nanotechnology for evil.
Wow.
But I also think that viruses are nanotechnology that's quite potent.
Yeah.
And I think that the sort of technology we're developing will also be able to mount very good defenses against those sort of things. And I think that there is a huge defense component to this because, immunological reprogramming to specific characteristics is something that we will be able to do. And
you can create massive firewalls inside your body.
Yeah.
You think you can create a firewall around all of your human cells so that way bioterrorism can't happen?
Well, I think it's always hacking and counter hacking, like just like we have right now, right? You're not going to get to iOS 9.2 and then hey, this is it. It's got to keep updating. So I think that now we have different selection pressures as humans, as just evolving organisms, where we have this technological component now that's entering and actually pushing our evolution, right? And that's something that can be used for good or for evil, which are very relative terms. But
Ron, yes,
yeah, evil. Funny.
Being evil is underrated. Just kidding.
You know what?
Is this with $150,000 to start for delivering information to the right address, and then over time hopefully getting that down to $1,000 for these gene therapies?
Yeah.
Do you think that this could exacerbate wealth inequality?
Always, and that's a big component. I think figuring out how to provide healthcare for people who don't have the money to pay for it is something that I'm really excited about, frankly, because if I can bring my cost of goods down to $100, then I think a billionaire should pay a lot of money to get a gene therapy because they're a billionaire, but that should also be subsidizing 100 people, right?
Yeah, who don't have.
Right,
exactly,
and I think that as companies we have to take stances on these sort of just dilemmas and issues pretty early on, saying, okay, we need to build towards this, right? And that's
conscious capitalism.
Yeah.
We just had a meeting with the CEO of Conscious Capitalism, Alexander. He's going to come on the show. That's huge. The fact that it's built into your values at such a young stage is super indicative of now going out and actually doing that and making sure that actually happens. And getting somebody on board, you're paying $10,000 so that you can subsidize, 100 other people. So that's boom. That's so cool. Okay. How about what is something that you believe to be true that almost nobody else believes to be true?
That biology is something that can be highly programmable, and that.
But I think other people think that.
But that I believe it in. There's Dave Asprey biohacking, and then there's cell-specific genetic engineering, grass-fed butter versus. Sorry,
Wait, what?
All I'm saying here is there's biohacking, and then there's precision engineering of deep biological traits on a deterministic level where it's like you now possess this trait or you no longer have this trait. And I think that's not something that people really grok yet on a high level. I think a lot of people are into biohacking, or they like the idea of gene therapy, like can I get different colored eyes or get different colored hair or something else.
Yeah.
But like that's where I think we need to teach people this language of different cells having different addresses and essentially start thinking about things that way, because that's where the next breakthrough happens. That's where like biohacking becomes, wait, we're actually biohacking.
Yeah. Okay. Now I'm more following.
Yeah.
I still think more people think that way maybe than you think.
I hope so.
I hope so too.
Out here, yes.
Yeah, out here, yes.
Yeah, I think globally, maybe not.
Yeah.
Right?
Yeah. How about? Do you think we're alone in the cosmos?
Oh boy, I doubt it. What does Neil deGrasse Tyson think?
I'm almost certain he thinks that there's 1 out of every 5 stars has.
Isn't he back on the God thing? Didn't he? Did I read that he endorsed the God theory?
I don't think.
Okay,
I endorsed the God theory, but I know that he's had a bunch of people on his shows recently that are religious, and I think he's trying to bring the conversation up at least more. Because God doesn't necessarily just mean religious God. God means the cosmos in general, the universe in general. There's,
Well, that's one understanding. Then there's something among us that wants us to believe it's God, which is also trying to figure out what God is as well. Now that's something to think about and ponder. It goes to it's all about the God and the devil thing, and then you have to be real careful. Everybody wants to talk, till the cows come home about God, but you start talking about the devil and everybody shuts up.
That's true. Yeah.
Sorry. Go back to, what was the question? Are we alone? Yeah. Yeah, go ahead.
And okay, so then how about, are we in a computer simulation? What do you think?
I think the computer is overrated. I think it's a Silicon Valley hype cycle. But what is a simulation, right? And what is a computer? So.
So then is the question. So then, what is your answer?
Well, I think. My answer is multidimensional. If you could model the whole universe and truly understand both directionality and position of every single bit of resolution within that universe, then in theory, at that level, you'd be saying, okay, well, this is like a simulation because I actually have modeled all this and I know exactly how it's going to behave, and everything is just like physical constants and somehow accounted for. But I also don't think that's how the universe works. I think the universe is like a highly parallel, kind of tumbly mess of parallel splitting off realities into many things, or even if it's one reality, like it's still so convoluted and so complex that to me that's almost the less interesting question than can we program universal constants.
That's cool.
And that's where the answer is yes.
Yeah. But then if we actually live in one, if we can tap into understanding that better, it'll awaken us. But yeah, I think so too. Can we program every single plank, every single vibration?
And how much of that is dictated by our consciousness just like coming up with the concept of creating something, that requires you to look around you and see that something doesn't exist. And your mind's like, I want that thing to exist. And then you will actually change your behavior and usually the behavior of other people also to create something where something didn't exist. That's pretty crazy if you think about it.
Yeah. And then, do you think that what is consciousness?
Oh boy. What is consciousness? I mean, consciousness is a state of being, and being aware that you are being. Maybe.
Okay.
Presence.
But do you think what do you think is the most beautiful thing in the world?
Most beautiful thing in the world. Being able to be in the moment and be peaceful. Like actually just being,
Yes.
right? Like those moments, really great. Doesn't matter where it is. It's like in your mind. We'll be releasing some just like white papers and educational materials on, I think that's really a goal of, one of my goals too, and the goal of our company is to educate and just help people to understand what's going on, because it's such a complex set of converging fields.